Hello and welcome to X-ray Crystallography for Dummies, which is a video resource created on behalf of your student team. This should be a really concise run-through of everything we've covered so far in the X-ray crystallography part of the course, and hopefully this should link together everything we've covered so far in lectures. To create a protein crystal from a solution of protein, there are some minimum requirements, and the first of these is purity, which basically means the percentage of the protein in the solution, that is the protein you want to look at. The minimum for this is generally 95%. Concentration is required to actually get the protein to crystallize out of solution. If that's too low, then you won't even form a crystal. And you'll need sufficient amount so you create a protein crystal big enough to actually run the experiment on. These values generally should all be as high as practically possible to get the best possible results, and the minimums vary depending on the protein. The problem we have with crystallization is that as more and more protein comes out of solution and becomes a crystal, the solution contains less and less protein, so it's less concentrated, which means that crystallization is less likely to happen, and after a certain amount it will stop. The way we get around this problem is to have the protein in a solvent such as water and in a precipitant. Within a sealed system you'll have the protein in solution, and you'll have a reservoir of precipitant which is a much higher concentration than in the protein solution. Because the solvent's volatile, it can move between the protein droplet and the reservoir of precipitate. Entropy is keeping the solvent in solution with the protein, but the disequilibrium between the two solutions is also forcing it over to the reservoir so that it can help even out the concentrations of the precipitants. This means that as the protein crystallizes and the concentration of protein in the protein solution goes down, the solvent is more free to move over to the reservoir of precipitant so that it can help even out the concentrations. And since this is happening, it means that the protein solution is becoming more concentrated again and crystallization can continue. So this is basically an equilibrium between the two solutions that allows crystallization to go on for a lot longer. This is just one example of how it can be done, and there are plenty of other methods for doing the same thing. Different proteins crystallize better under different solutions, so depending on the protein you're trying to crystallize, you might want to use a different temperature, a different precipitant, or maybe a different method. To find out how best to crystallize a protein, it can be a good idea to run lots of mini tests to find out which parameters work best for crystallizing that protein. Because protein solutions are really expensive to make, you generally get machines to run lots and lots of mini tests to see which parameters work best for crystallizing your crystal. For these mini tests, you don't have to have as much as you'd need for an actual X-ray crystallography experiment. Machines tend to be better for this than humans because they can pipette smaller amounts more accurately. So now that we know how to make a protein crystal, we're going to have a quick look at the structure of the protein crystals themselves. Proteins themselves have a really irregular structure, but in a crystal lattice they're very regularly arranged. Because proteins don't tessellate, this means that there's huge gaps within the structure. These gaps tend to be filled with whatever other molecules are in solution with the protein at the time. And some of these molecules are always arranged in the same way, so they're quite specifically bound to the protein non-covalently, and some of the proteins are just floating free in solution. So this means that the rigid small molecules come up with the protein on the X-ray crystallography trace, and the random ones don't. Having all these gaps means that the proteins are less strongly bound to each other, which makes it harder for a protein to crystallize than other molecules like salts. And this is why the crystal structure is so hard to make and so unstable. We're going to have a look at the three terms you'd use to describe a protein crystal, which are the unit cell, the asymmetric unit, and the space group. The crystal structure is like a big repeating 3D pattern, and a unit cell is the smallest possible block that you could take out of that pattern and place it one on top of another to create the pattern as a whole. By definition, there can be no gaps between unit cells, and unit cells have straight edges and straight sides. If you took all the bits of protein from inside a unit cell and put them back together, you should end up with a whole number of proteins. The red shape on this picture is an example of a unit cell of a protein, and the protein's there in yellow. And as you can see, when you add more identical red cells next to it, the contents of each red cell are identical. This is just a side-on view, so you're only seeing the protein in 2D, but if you were to get a 3D view of this protein, you'd see the unit cell as a big block, and that block would still repeat horizontally and vertically. So going back to the previous slide, we can say that the unit cell describes the shape, size and position of the smallest repeating 3D block in our model. The asymmetric unit's very easy to describe. It's just the exact shape of our protein and its bound ligand, which is what we're trying to find out. Protein molecules in a lattice have rotational symmetry around different axes, and the space group is how this rotational symmetry is described. This example is P222, which is a good one because from whatever side you look at it, it always looks the same. On the diagram, the lines indicate axes of symmetry, and these are axes of rotational symmetry, so instead of planes, they're lines. And the little red circles indicate the positions of protein molecules in that lattice. So now we're going to break this space group down into the letters and numbers that describe it. Letters are used to describe the general shape of a lattice, so where these axes of symmetry actually lie, and P just means that it's a non-centered lattice. And those points you see where the lines intersect, they do not stand for proteins, they're just literally there to describe where the axes of symmetry cross each other. The first number describes the rotational symmetry around the first axis, and the second axis, and the third axis. So if you look at the first example of Kermit the Frog, you can see that he's rotated 180 degrees around a line going from left to right, so that in a whole circle you'd have two of him. If you look at the second example on the right of Fozzie, you can see that he has fourfold rotational symmetry. So within a whole circle there are four of him at intervals of 90 degrees. For Fozzie you'll have to imagine the plane actually going into the page. So that's simple rotational symmetry, and a number you have indicates how many of them you'd have rotating around a plane in a full circle. With screw rotational symmetry, as well as rotating around a plane, each rotation is staggered so that one's moved slightly further up by the time it's rotated round. On this page we have Gonzo demonstrating simple 2-1 rotational symmetry. So he moves around 180 degrees and he's moved along a little bit more each time he does that. On this slide we've got a view of P222 from one side. The lines again represent planes of rotational symmetry. The R's represent proteins, and those dots simply represent the places where these planes intersect. At each dot, try to imagine another line also intersecting that's going into the page. The bigger, darker R's represent proteins that are slightly closer to you from the side you're looking at it, and the smaller see-through ones represent proteins that are slightly further away. So now, if you focus your attention on one corner of this diagram, you should hopefully be able to see how around each plane the R rotates. So on the axis going from left to right, around there you should see that one R is the right way up and one R is upside down, so it's rotated along that axis to become the other way around. And on the axis going from up to down, you should hopefully see that the R has rotated so that now it looks like it's going the other way, like you're reading it from right to left instead of left to right. Now if you look at one of the big R's or one of the small see-through R's, and focus on the other R which has a red dot in between that and it, you should hopefully see that that R has not only rotated so that it's upside down, it's also facing the wrong way, and that is rotation around the axis going into the page, so the third axis. So going back again, we can say that a space group is a description of how proteins are arranged in the lattice in terms of plane positions and rotational symmetry around them. When you actually get round to firing X-rays at your crystal, you can have your crystal at room temperature or frozen. When X-rays interact with water molecules in the crystal structure, what they sometimes do is split it into radicals, and these radicals can react strongly with your protein and affect its structure, which is something you don't want. One way to get around this problem is to freeze your sample. This means that the water molecules have less energy, So when they're split into radicals, those radicals have less kinetic energy, so are less likely to react with your protein, just like freezing something will slow down the rate of any chemical reaction. If you do this, you have to make sure you freeze your sample very quickly using liquid nitrogen. And this is because if you freeze water slowly enough, you form ice. And since ice is also a crystal, it would have its own diffraction pattern. To generate your X-rays, you'd normally use a synchrotron because this allows you to select a wavelength. And this is good because the wavelength you use is quite a big factor in determining the resolution you get. And wavelength is also useful later on in phase determination, which we'll get to. This is a very simple diagram of what actually happens. Obviously your crystal won't look like that weird green line, it will look like a crystal. So you fire X-rays at your crystal and you measure how they diffract. And in order to gain enough information to work your way back to a 3D structure, it is necessary to rotate your crystal so that you can get a view from every angle. Thinking about a single molecule, when X-rays pass through it, the electrons in that molecule change the direction of the X-rays. These diffracted waves can then interact with each other, and they'll produce a new set of waves, where there's been some constructive and some destructive interference. If you were able to record the diffraction pattern from a single molecule on X-ray film, you'd see large areas of both high and low X-ray intensity. You'd also be able to see intermediate intensities as well. Obviously it's not possible to isolate and run an experiment on a single molecule, and that's why we have to use crystals. Because molecules in the crystal are regularly arranged, and all the X-rays are coming from the same direction, that means that if a particular part of a particular molecule diffracts the X-rays in a certain direction, then that effect will be duplicated by other molecules in the crystal. If the same part of the same molecule is diffracting waves in the same way on two parallel planes, then there could be constructive interference between the waves produced, which would amplify their effect and make them visible on the X-ray diffraction pattern, if the effect was repeated along the whole lattice. The wave has to travel further to diffract off one molecule than it does to diffract off another. So the factor that determines whether they superimpose or whether they cancel each other out, or some intermediate, is whether one had to travel a whole number of wavelengths further than the other so that they're still in phase. The wavelength of the X-rays and the gap between the two planes is important in determining whether or not the diffracted waves will be in phase. Also, what's important is the angle of the incoming X-rays compared to the plane, and the X-rays will be diffracted off at this angle as well. This means that crystals are very selective in the angles at which they diffract, which is why you get a discrete pattern of spots on your detector when you're looking at an entire crystal. So when you've got a diffraction pattern for an entire crystal, the angles of the diffracted waves, those spots, are determined by the structure of the lattice, and the only piece of information that you can link to the protein itself is the relative intensities of these spots. Something you have to take into account when you're working back from diffraction data to a lattice structure is that not all the proteins are in the same orientation because they're undergoing symmetry. Some of them are upside down or the wrong way around. Nowadays we know the kind of diffraction data produced by each possible space group for a protein crystal, so it's very easy just to compare things to previous experiments using software and to have your space group worked out pretty much automatically. The mathematical method used to derive a protein structure from diffraction data is called the Fourier transform. The Fourier transform works back from the complex waves that reach the detector to the smaller waves that were diffracted by the original molecule and actually make up these complex waves. And from those smaller waves, it then works out the electron densities at different points in the molecule. And this method requires three pieces of data: the angle of diffraction of each of these waves that reaches the detector, the amplitude of the waves that reach the detector, and their phases. The angle of diffraction is known just from where they are on the detector, and their amplitude is related very closely to the intensity of the points. The difficult part of that is working out the phase because we can't measure that directly. The first ever method used to try and work out these phases is called multiple isomorphous replacement. It involves crystallizing your protein while it's in solution with high concentrations of heavy metal ions. The metal ions bind to some of the proteins at specific points and become part of the crystal structure. In order for this to work, the protein with the metal ions bound has to have a very similar conformation to the protein without the metal ions. Diffraction data is collected from the protein without the metal atoms and then with the metal atoms. Because the metal atoms are such large areas of high electron density, it's very easy to separate their part of the diffraction data from that of the rest of the molecule. Because metal ions produce such clear diffraction data, and because there'd only be 2 or 3 of them in the molecule, it's possible to estimate their phases just by plugging in possibilities of where they could be and seeing what the diffraction data for that would look like. This is helped by the fact that when you do your Fourier transform, you have the constraint that all electron density has to be either very great or very small, because unless you're in the electron field of an atom, there is no electron density. Also, if you get any major negative values for electron density, you know this position is wrong because that's impossible. Once you know the phases of the complex waves produced by these atoms, it becomes a lot easier to work out the phases of all the complex waves produced by your molecule, and you can use these as a sort of road marker to working out the others. You've also still got the constraints of having to have very high or very low values of electron density, and only to have positive ones. Multiple anomalous dispersion is a lot like MIR. The difference is that you choose wavelengths of X-rays that these metal atoms that you incorporate will absorb. This will create holes in the diffraction data, and by observing the link between how we change the structure of our molecule and how we change the diffraction data can again be used as a way to work out the phases of the complex waves. When using this method, we sometimes incorporate selenomethionine into our molecule, which is methionine but with selenium used instead of sulfur. The general principle with this method is the same as MIR, in that you're changing the structure of the protein, and you can work out the phase by observing how that changes the diffraction pattern. The third way to work out the relative phases of your waves is by a method called molecular replacement. This involves taking the diffraction data from a very similar protein with hopefully the same conformation. If your protein has over 20% sequence identity to another protein, they will probably be of the same conformation. You can work out the lattice structure of your protein without phase information just from the relative positions of the dots. If the 2 proteins you're comparing have different lattice structures, then this can be accounted for. The relative phases of the waves diffracted from the other protein provide a very good starting point for your phases. You can then continue to refine these phases until you have a diffraction data that fits a good chemical model of your protein. So this is just an overview of how structures are worked out. So first you look at the pattern of the dots to work out the lattice structure and therefore the space group. And then you look at the intensities of the spots. And this part's more complicated because although you have these techniques which can give you some estimate of the phase, there's a lot of trial and error and trying to find data which fits both the diffraction pattern and the phase information. Once you have a model, you can also get these statistics which tell you how good it is. Resolution is a measure of how far apart 2 points have to be before you can distinguish between them. Reducing the wavelength of your X-ray helps you get, in theory, a better resolution, but in practice the resolution is always determined by the quality of your crystal and the crystallography experiment, so it will only help if it's been a limiting factor. The rest of these statistics are fairly self-explanatory, and there's more detailed information on them in the online resources.